let me just introduce Jim Faeder from the University of Pittsburgh, modeling the dynamics of coronavirus and alpha virus infection. Of course, Jim has been working on viral replication modeling for many years and also develops very nice software tools. And we appreciate his being willing to give us an update on his work.
Thank you, James. Okay, I share.
And I'll warn you at 5 minutes as well.
Well, thank you. I will try to be quick and brief. It's not true that I've been modeling viral replication dynamics for a long time. I'm fairly new to this. And I just wanted to give you a little bit about my background before starting out here. So my group is largely interested in modeling cell decision processes, and we use a method called rule-based modeling to do that. So we're really interested in what goes on in intracellular signaling networks. And with the goal of eventually modeling drug-protein interactions and learning how to control the outcome of signaling processes and to direct them towards therapies. We use an approach called rule-based modeling that I'll describe a little bit in the next couple of slides, just to give you an orientation, again, on the kinds of technologies that we like to use. And I think they can be fruitfully brought to bear on problems in viral dynamics, which is something that I'm really interested in doing moving forward. But as you'll see, our results on that are a little sparse at this point. The software that we developed to do rule-based modeling is called BioNetGen, and we develop and maintain that software. And I'm hoping to get to the end to show you some cool new stuff that we're doing in terms of interfacing BioNetGen to make an integrated environment for modeling. So just briefly, the challenges that we see in modeling cell regulatory networks, and I think they're very similar to the kinds of challenges that you see in modeling intracellular replication dynamics of viruses. As many of you know, I'm sure, the molecular details of these processes are extremely complex and ones that have been rather sparsely modeled. Most of the models that have been developed of viral dynamics are really at the scale of very, very coarsely treat what happens inside an individual cell. So we're interested in trying to develop a finer grained version of some of these models. And I think we can fruitfully use some of the methods that we've used to model signaling proteins. And there, the challenges are similar, that signaling proteins have multiple sites of interaction. They're modified in multiple ways, and they can interact with many different other proteins. And this gives rise to a phenomenon that we call combinatorial complexity, which can be illustrated simply as, if you have even a small number of proteins that interact with each other and a simple set of interaction rules, the numbers of ways that they can combine and give rise to chemically distinct species is combinatoric and results in sometimes very large and complicated models. Those models are characterized by relatively small number of parameters compared to the number of actual constituent species. And so they should be able to be concisely described in a model and simulated, but the standard tools writing models as differential equations don't scale well for that problem, because of that problem. And so rule-based modeling provides a flexible and compact way to represent biochemical interactions. So the core, sorry about this spasming here. The core concept is that the biological molecules can be abstracted as structured objects. So instead of writing a species just as a name of something, we represent it as a molecule with different components. Those components can bind to other components of the same molecule or other molecules, and they can also be have different conformational or post-translational states. So in this way, we can we have a representation for potentially complex biochemistry, and then the rules are used to define molecular interactions. And a simple example rule is a domain of a particular protein. Here it's a tyrosine kinase. Binds to a phosphorylated region of another protein to form a bond and to form a complex. And the key concept that underlies rule-based modeling is this idea of don't write, don't care. So these proteins are multifunctional. They may have many other interactions with other proteins, but the interaction between the kinase and the receptor doesn't depend on those other interactions in the sort of coarsest level description, and we can write a single rule that captures that interaction. Typically, we describe interactions by mass action kinetics or some form of generalized rate laws that can incorporate more complex biochemistry. Okay, and so really the underlying idea is that we can bridge then from molecular scales of individual proteins and their interactions with other proteins all the way up to this network scale. We do this by abstracting the structure of proteins to have these components that can interact with other proteins, and we can build up complex models of large-scale networks. I mean, this way, and here I'm representing it a large network model of EGFR signaling as what we call a contact map, which just shows the proteins and sites that have been modeled. And a key point about this contact map is that it contains the complexity of the rule set, but the underlying species complexity is much greater, and that's handled either by an automatic network generator or by simulation algorithms that don't depend on that complexity explicitly. The BioNetGen framework that I'm describing and standard, there are many other tools for rule-based modeling as well that I'm not talking about here. But the key idea is that the molecules and interactions are represented in a concise form as a set of rules that can be passed to an interpreter that then either generates all the species and reactions that are implied by the rules, or can carry out directly Monte Carlo simulations based on the dynamics that are described in the rules. And at the end of the day, we generate time courses of dynamics. We can generate steady states as well. And then we use those time courses, we compare those to experimental observables. So, exactly the same kinds of rule-based interactions that we use to describe host signaling network can be used to model the viral dynamic component as well, and in particular also the interactions between viral proteins and other viral components and the host signaling pathways. So we got interested in modeling the dynamics of SARS-CoV-2 infections like everybody else to see if there was something that we might contribute. I've for a long time been interested in the idea of how viruses interact with and co-opt host signaling and other intracellular machinery. But this was really the impetus that got me to go in and actually try to do this in a somewhat systematic way. And so I found out about the effort that was being led by Paul Macklin, and I think Paul has given a talk already in this series and introduced some of you who may be here now to the PhysiCell framework that he develops. It's also, I met James Glazier in the context of this modeling effort as well. And the goal was to develop multiscale model of COVID-19 dynamics spanning from the subcellular level all the way up to the whole individual level, spanning these different cells, what goes on inside of a cell, how cells interact with each other, the multiple different components of both the innate and adaptive immune system that come to bear on infections, spread of cytokines, and other factors in the host tissue, and then ultimately how different compartments of the tissue might interact with each other during the spread of an infection. And the goal here, it really is to bring into a dynamical framework the full scope of the host pathogen interactions and immune response, with the goal of being able to then test different therapeutic strategies, and also to understand basic aspects of the infection dynamics, which really are much more complex than we've been able to model. So Paul did a great job of bringing together this international group of collaborators to develop this SARS-CoV-2 tissue simulator using his PhysiCell framework. The consortium has involved up to 40 or so people at 20 different institutions. I'm not sure how accurate that count is now. I mean, it's grown over time. And bringing together lots of people, both who are very experienced in these areas and people like myself who are relatively new. And in Okay, so yeah, I should mention just briefly that the PhysiCell framework is an agent-based framework for modeling multicellular interactions. Its primary application up to this point was to model tumor growth and dynamics, but Paul's very keen to bring it to bear on viral infection dynamics as well, and that's what the focus of this effort has been. And so it's really been a team science approach that his group has been leading, bringing together people from very different backgrounds and disciplines with the using open source tools to develop this simulator and to try to do it fast and in a time when obviously we can't get together in person and we have to rely on all the different digital virtual tools, both for in terms of human interaction and in terms of code development. So that's all been great. I'm going to focus the rest of my time here just on talking about the model that's been developed and a little bit of the preliminary analysis, just to give you a flavor of really some of the things, the places that we would like to go with these kinds of approaches. So the initial version of the model was developed primarily by Paul and people in his group in discussions with others, and was available before I even became involved. So the core framework for describing the infection of a tissue was worked out before I got involved. But and then has been rapidly developed. So first we had just individual cells representing epithelial cells in a tissue that had a very simple set of modules that described the process of endocytosis and then viral replication within the host cell. And that enabled them to carry out different kinds of simulations under different conditions to identify the at a very qualitative level the effect of modifying different aspects of the simulator. And one thing I can emphasize at this point was the goal of the rapid prototyping was to develop the framework for models that had this sort of the correct underlying coarse-grained physical descriptions, but they models have not been calibrated to experimental data, and that's meant to be left to the domain of people who adopt models or subsequent efforts of the overall group. The goal here is to develop physically realistic descriptions. So the immune response team group of this consortium has been very active in the months, I guess, in the past summer, primarily developing quite an intricate model of the host immune cell response to infection, and built out the model to a much greater range of interactions and complexity. And I just show a few time courses here. I don't know if some of the others who were involved have already given talks. I know they have done a lot of additional analysis beyond what I'm presenting here. My group was primarily involved in the intracellular dynamics modeling, and initially my goal was to take this very simplified version of viral replication, which treats this more or less as a linear cascade of events, and to make something a more fine-grained molecular description. So far, we haven't made tremendous progress on that, in part because there's not a tremendous amount of experimental data to support that kind of effort just yet. And so part of what we've been doing is trying to find collaborators who are willing to do the kinds of experiments that could inform such models. And in the meantime, we've learned a bit about how to model these systems. First thing that we did, of course, was to translate it into a language that we like to build models with. And we wanted to explore, in particular, developing the capability to make our rule-based models and to export them to create modules in this larger multicellular agent-based framework. So I'll mention a little bit about our experience in doing that before I wrap up. Some key properties of this model: it replicates very nicely. This lag phase during which following the initial infection of the virus to a cell prior to the export of virions to the extracellular environment. And one thing that I noticed very early on in working with this model was that the number of exported virions did not match very well with what was thought to be the burst size, the viral burst size, which is estimated to be in the hundreds or even a few thousands of virions per cell. And so, you know, the immediate question that came up, and I recall having multiple discussions with James Glazier on this point early on, was how does the virus amplify its RNA, and how does the infection of the cell by one virus result in the production of thousands of virions? So it was really necessary.
5 minutes.
Okay, good. I'm doing okay, I think. I put the stuff that derails me that I love to talk on and on and on about at the end, so that's good. We, you know, having talked to James, and they published a paper, and I should mention that, you know, the CompuCell 3D has been used to build a very similar kind of multicellular agent framework for modeling the infection dynamics. A key step is that there needs to be an amplification reaction in this that amplifies the production of the full-length viral RNA, and putting that into the model enables there to be amplification and enables us to then much better fit the time course of replication. It sort of matches much better the observed viral titers from cell culture experiments. And it's actually interesting, you know, there's still a fairly limited amount of data about the viral titers in various different cell culture systems. So I don't know that there's been a precise measurement of actually the burst size in SARS-CoV-2 up to this point. There hadn't been last time I had checked. And in fact, our collaborator has done some measurements on that, which I won't present in this particular presentation. But what I wanted to do here just is to show briefly that, you know, that we can learn something about the model by doing a sensitivity analysis. And here we did a regression-based sensitivity analysis to try to figure out what are the factors that of the, you know, the parameters of this model that strongly correlate with the experimental burst size. That seems to be something that's highly variable in different, for certainly different viruses. We saw that there's almost a factor of 2 orders of magnitude difference in the viral loads produced by SARS-CoV-1 and CoV-2 in the data that I showed a little bit earlier. So we were interested in trying to identify what are the key steps in viral replication that might lead to different burst sizes. And as I mentioned, we used this regression-based approach where we did multilinear regression on the with respect to the model parameters on burst size. I should mention we did this in the log parameter space, and we're able to find parameters that correlate with a change in burst size over around 6 orders of magnitude. And they're sort of what you'd expect based on the amplification step being, you know, a key factor that controls the total virions that are produced. So the parameters that are in this middle part of the, you know, where we see large sensitivities are exactly the ones that we would expect. But our goal was to first to do this as a sort of a proof of concept for the single cell model, and then to to be able to do this kind of analysis on the multicellular model and understand how these factors play up at the larger scales. So I had a few slides here. I'm going to go very briefly through this just to say that one of the areas that I think we identified as over the course of working on this collaborative project is, you know, we weren't able to just plug in our BioNetGen models into the framework in a very convenient way. And this actually, the fact that this all has to be, the integration has to be done by hand coding at this point, you know, proved to be time consuming, and also, you know, is somewhat error prone, and something that I think is a target for hopefully, you know, future development of such frameworks to address. But yeah, so this has been a time consuming process to try to make sure that we validate that the implementation of these models matches the original description that we had encoded. Once we have the integration working, which it largely is at this point, we can then, as I mentioned, do sensitivity analysis on the larger model. And this is really just a preliminary result, but to show that, you know, even with this regression-based method is pretty sensitive. So even with this relatively small numbers of samples, we're able to identify model parameters that correlate very strongly with changes in for example, the burst size, so measures of infectivity. We're currently in the process. So one of the things that we were encouraged to do is by, as part of working on this project, was to find experimental collaborators. And I have an expert viral dynamics modeler in my neighborhood as well, who I've teamed up with, and we're now co-supervising a student who's working on modeling different plus-stranded RNA viral kinetics. That's Caroline, who's listed at the bottom. We're working currently to refine this replication model and incorporate more precise measurements of the viral kinetics, and ultimately time courses that contain measurements of other important cellular responses, such as, for example, interferons at the same time. And ultimately, the goal is to develop integrated models of the cellular host response that involve interferon signaling, and in particular, the interaction between the host and viral replication, and how that can be shut down both in an intracellular fashion, but also in a paracrine, autocrine, paracrine fashion. I mean paracrine and something else fashion, but okay. We also, another thing that I think is of interest is, our collaborator is an expert in studying of alpha virus, which is a nice model organism and a nice model virus that has a much simpler genome, and is also of health relevance, but in particular for biodefense efforts. These alpha viruses, a number of them cause very lethal forms of encephalitis. And there's a lot of interesting interplay between the virus and the host that leads to modulation of interferon signaling that occurs not only within individual cells, but in a paracrine fashion, but also over longer distance scales. So, for example, infection in the periphery of a mouse, so in the mouse foot pad, for example, if cells are triggered in the right way to produce interferons that can be protective for against the encephalitis, infection of the brain subsequently, and these number of alpha viruses have developed pretty sophisticated means to evade immune detection near the site of infection or in draining lymph nodes that prevents this from happening. So I'll stop. I'll wrap up there and acknowledge all my collaborators I've mentioned as going along. A plug for various modeling tools. We have our own multiscale modeling center at the University of Pittsburgh. This thing called MMBios, and we're focused largely on the intracellular scale. So going from proteins up to spatial and simulations of intracellular dynamics. And so I think we have a lot of nice computational tools to contribute to viral modeling efforts there. I wanted to put in a very short plug for a recent extension we wrote for VS Code that I think provides a very nice integrated environment for modeling and for connecting proprietary modeling languages like the BioNetGen or special purpose languages like BioNetGen to code development efforts based in Python and other languages, which VS Code provides a nice environment for. The modular structure of this system has let us sort of replicate the integrated development environment we had previously in Eclipse. We can run simulations and make plots. We can export Jupyter notebooks and make models accessible to various different frameworks in a very straightforward way. So we're looking forward to further developing those capabilities. And I think this is a place and a framework where a lot of our multi-scale efforts could be supported nicely. And with that, I will thank you for your attention. Happy to take any questions.
Okay, our meeting time is over formally, but we have a half an hour for breakout rooms. Now, I hope our speakers and our audience, those of whom wish to ask questions, will attend.